everybody, and welcome to Chapter 1. Chapter 1 is just going to be an overview or introduction to biology. We'll run through some basic characteristics of living things, evolution, levels of biological organization, the scientific method, and critical thinking skills when it comes to scientific claims here. Looking at this image here, looking at this next slide here, all living things are made up of 4 macromolecules, and the term macro means large. So these are basically our 4 major components that make up our body. This includes nucleic acids, proteins, carbohydrates, and lipids, and this will be discussed more in the chemistry chapter. Looking at nucleic acids specifically, this is the backbone for our DNA and our RNA. Our DNA is our coding system found in all cells of our body. Most of it is found in the nucleus of the cell, and it gives our cells instructions on how to make proteins. Now, proteins are long chains of amino acids, and our DNA gives us the instructions to make those proteins. Carbohydrates, these can range from simple sugars to complex sugars, very large sugars called polysaccharides, and these are used in a foundation for making things like ATP. Lipids, triglycerides, these are a source of energy, or they are a source of hormones, like steroids can make hormones. They're also components of our cell membranes. So all living things are composed of cells. This is the cell theory. And the cells are the smallest units of life, and they have arisen from other pre-existing cells. They are able to grow, and they are able to replicate. They are able to reproduce. They're able to use energy and to carry out metabolism. Now, metabolism is the chemical reactions that occur in cells. And we will talk about all those chemical reactions throughout the next couple chapters here. All living things have response to their environment. So if you have a pinprick to your arm, you feel that there is pain. If you feel heat, if you have a light shine in your eye, you blink. So living things, they respond to their environment. Not everybody responds to their environment in the same way, but they do respond to the environment. And they try to maintain homeostasis. Now that can range anywhere from temperature homeostasis. For example, if you're cold, you would activate your muscles to start shivering. The shivering of those muscles would cause a byproduct of heat. If you're hot, you sweat. That would cause the vasodilation, meaning increased blood flow to your skin, and that would cause blood flow to reach to your surface, and then heat would leave your body via evaporation. Now, we also have other things inside the body that can respond to homeostasis. For example, there's pH levels. Certain parts of our body need to be maintained at certain pH levels. So we'll secrete substances to maintain that pH. Over time, we have adaptive traits. We've gone through evolution, and part of evolution is adaptation. And so this allows us to survive and reproduce. So this goes back to Charles Darwin in Survival of the Fittest, and that we have adapted to our environment. We have adapted to our environment. So whether it's changes in the amount of hair, changes in our height, or changes in the way that we act, all of this has allowed us to survive till today. Now, over time, we have evolved. An individual does not evolve. I myself am not going to evolve in the next 10 years. I'm not going to all of a sudden grow hair in thicker lengths over my arms or grow another 6 feet unless I had some sort of disease. So it's a population that evolves over time. There's various animations in your lecture, so this is not opening for me, but when you go through your lecture slides, this would be available for you. All right, so like I had said, the term evolution, we have evolutionary relationships. There are things in the human body that allow us to correlate to other animals, such as, they're called vestigial structures, such as we have a coccyx, which is part of our tailbone, and other animals also have a tailbone. And so organisms that we're closely related to, we have more similar characteristics. For example, of the mammals, we can share diseases. We've all seen that as COVID-19. It can transfer from animal, or SARS. It can transfer from animal to human. And we kind of organize everything into 3 domains. So all of the environment is organized into 3 domains. We have domain bacteria. We have domain archaea. Those are one cells. We as humans, we're multicellular, so we belong to domain eukarya. There's various differences between these. We'll talk about this in the cell chapter. But for example, in the domain Eukarya, this includes a wide variety of kingdoms, including the Protista, fungi, plants, and Animalia. We have membrane-bound compartments, membrane-bound nucleus, membrane-bound organelles. And so we have a little bit more complexity than bacteria or archaea. We have a relatively large brain as humans. We stand up on 2 legs and we have opposable thumbs. This sort of separates us from other animals. Going through the levels of biological organization, I'm going to go right to this picture right here. And so we're looking at the smallest unit of life. We have what's called atoms. Atoms will come together to form molecules. Molecules will make up what's called our cells. Our cells will come together to make our tissues, like our bones, our muscles, our skin. Those types of tissues come together to make organs. Organs come together to make organ systems, such as like the GI system, the cardiovascular system, the skeletal system. And those systems make up the organism. Now, the organism can live amongst other organisms of the same species, and that would be a population. So all the humans in McHenry County is a population. Now, the humans plus the birds plus the coyotes, that would make up the community. Now, the humans plus the birds plus the coyotes plus all the rocks and all the lakes, that would be the ecosystem. And within the whole biosphere, within the entire Earth, that would be the biosphere, including land, water. All right, so that gives you a organization of your levels of biological organization. All right, so the next thing that we have is the scientific method. And the scientific method is kind of like how we prove something is true. And you'll find a great description in your lab book in the first lab. It's at the end of the first lab about the scientific method. And so science is how we answer questions of thought. We're trying to explain something. So for example, I think that people who take vitamin C, they're less likely to get the common cold. So I'm going to do an experiment. And I'm going to do an experiment following the scientific method. So it's a logical approach to how to take in information and determine if that information is true. You know, we do testing and so forth. And so you're going to first start off making observations and asking questions. So my theory about vitamin C, I think that people who take Airborne, you know, Airborne, you guys can buy that over the counter, has intense levels of vitamin C. I think that they don't get the cold as often. And plus, all the packaging and the labeling kind of tells me that too as well. So I'm going to ask some questions, you know, like, okay, so do you take this every day? How long have you been taking it? Do you only take it after you get a cold? Or are you taking it during cold and flu season as a preventative method? So then I'm going to form a hypothesis. And a hypothesis is basically an educated guess. So I'm going to say, all right, I think that people who take vitamin C are less likely to get the cold. And I predict that when you take vitamin C, there'll be a whatever percentage chance or you won't get the cold. And so that's the prediction that I'll make. All right. Now, I will create an experiment. Okay. And that experiment will either support or it will reject my hypothesis. And so let's just say I pick 2 groups of people. I picked 2 groups of people. They're both of the same age, randomly males and females, 20 people in group A and 20 people in group B. One group will be the independent variable. And so that is the one that's the factor. Okay, that's the group that's going to get the vitamin C. So one group is going to not get the vitamin C and one group is going to get the vitamin C. I'm going to take both groups and I'm going to put them in a room and I am going to blow in the ventilation system tons and tons of cold viruses. Sounds horrible. And then I'm going to take the data that I've gotten from my experiment. So I'm going to have them leave the room and I'm going to say, all right, report back to me in 3 days. Over the next 3 days, keep a diary of your symptoms that you may have. And so I'm going to come back. I'm going to measure for any fever, any nose running, any kind of cold-like symptoms. And I'm going to also ask for their diary over the last 3 days. And that's going to be my data. So I'm going to take my data, and then I'm ultimately going to reach a conclusion. Okay? I'm going to develop new questions if the conclusion didn't come true. Okay? With the conclusion, I need to look at problems like, well, the people that didn't get the vitamin C, or the people that didn't get the vitamin C and they got the cold, were they maybe around younger children? Were they exposed to more people in a population? So I need to ask new questions, and I may have to develop additional experiments. So I may need to keep them all quarantined for 3 days after I expose them. And so if it is true, and if it follows suit and all the data supports my hypothesis, then I could potentially come up with a theory. There's not too many theories out there, though. Okay, so this is just kind of a nice overview here. You have an observation, you form a question, you develop a hypothesis, you make a prediction, you experiment, you look at your data, that's your result, and you draw a conclusion. And you may have to restate your hypothesis or reformat if your results don't support that. Okay, so looking at, these are just some things from your book here that's talking about an experiment with LDL level. Sorry, I shouldn't have put that in here. There's this term called inductive reasoning and deducting reasoning. Inductive reasoning includes facts through observation until there is enough facts to draw a conclusions. So all organisms are made of cells based upon years and years of findings by biologists. Now, deductive reasoning involves making a general statement. If and then. If biologists have been saying that all cells, this previous one here, where do we go? If previously biologists have been saying that all organisms are made of cells, well, if you're an organism, then you're made of cells. So it's like an if and then. Okay, so inductive includes like doing the experiment. Deductive reasoning is based upon previous experiments. You can maybe make an assumption. Experiments that are dealt with medicines or materials or laboratory animals, they have to go through clinical trials, have informed consent. A lot of clinical trials we use what's called a placebo. So like I said, with my experiment with the vitamin C, I would give an airborne tablet to one group, and then the other group I would give a tablet that was the exact same color, the exact same taste, and the exact same size. A double-blind study, this is when both the patients or the test subject groups and the experimenter don't know what. So it's like, let's say my patients or my experimental test subjects, they don't know who's getting the vitamin C, and I don't know who's getting the vitamin C. So epidemiology studies, this looks at larger populations. So these are just out of your textbook there, some images out of your textbook. And so back to the last thing that we have with critical thinking. Critical thinking is used. This is when you kind of draw conclusions from your facts and your data. And this is really something that we'll do throughout lab. And so once you have this experiment and you want to publish it in a journal, okay, what's the word I'm looking for? A like medical journal, okay, not like Reader's Digest, okay? They're called peer-reviewed journals. So let's say I did this experiment on my vitamin C. And so this would be the Journal of Immunology Specialist, right? Because it's the immune system, right? And so before my paper, my experiment would get published in this journal, the top immunologist in the country would review this article and just basically make sure what I'm talking about is legit. So that's what a peer review is. So not everything online that we find is relevant or accurate, and everyone should evaluate where the source is. Okay, so that is it for chapter 1.